Good evening, everyone. I welcome you all to the live NPTEL problem solving session for bioinformatics algorithms and application course. So, this is for problem solving session on week 3. So, as the previous lectures in this also, in the initial part of the session, I'll provide summary of the previous week lectures, that is week 3 lectures. Then I have some sample problems will be displayed. Yeah, so before starting with this week, in previous week, we have learned about what is the basic structure of protein, DNA, carbohydrates and lipids, followed by we have started with sequence alignment concept. So in the sequence alignment concept, we have completed dot matrices and also have given introduction to the scoring matrices. So this week lectures are mostly about the scoring matrices such as PAM and Blossom, followed by we'll be handling how a dynamic programming alignment methods such as Needleman Wunsch and Smith-Waterman were performed. So we'll be take up some numericals on solving these two, followed by introduction to BLAST. So to start with, what are scoring matrices? In bioinformatics, scoring matrices are tables used to assign scores to the alignment of sequences, typically DNA, RNA, or protein sequences. These scoring matrices are usually used in alignment algorithms such as Needleman-Wunsch and Smith-Waterman. Scoring matrices are constructed based on the idea that certain substitutions, that is, replacement of one amino acid or nucleotide with another, are more likely than others due to evolutionary relationship or biochemical properties. I'm repeating again, scoring matrices are constructed based on the idea that certain substitutions are more likely than others due to evolutionary relationship or biochemical properties. The scoring matrices typically have positive score for matches and negative score for mismatches. These scores are used to evaluate the similarity between sequences and guide the alignment algorithms in finding the best alignment. Different matrices may have different scoring schemes based on the assumptions made during the construction and data used to derive them. Choosing an appropriate scoring matrix is essential for accurate sequence alignment and bioinformatics analysis. Scoring matrices such as Blossom and PAM are constructed based on the observed substitutions in homologous sequences. The scores in these matrices the scores in these matrices reflect the likelihood of one amino acid being substituted by another during evolution. Different matrices may be more suitable for aligning sequences with different evolutionary distances. By understanding the evolutionary relationship between sequences, that is, orthologs, paralogs, or homologs, one can choose appropriate scoring matrices and align the algorithms to accurately align and analyze the sequences. Additionally, knowledge of these relationships aids in interpreting the results of sequence alignments in terms of evolutionary history and functional conservation and divergence. So to start with the first matrix, that is PAM, which stands for Point Accepted Mutation Matrix, which is used to quantify the evolutionary distance between two sequences. It is used in the sequence alignment algorithm to score the likelihood of different possible alignments between two sequences. The PAM matrix is constructed using a statistical analysis of large number of closely related sequences. The underlying assumption is that mutations occur at relative constant rate over time, and the frequency of amino acid substitutions can be used to estimate the evolutionary distance between sequences. The values in PAM matrix are represented in terms of PAM unit, which represents the estimated number of mutations per 100 amino acids that have occurred during the evolutionary history of the two sequences being compared. So the values in the PAM are expressed in terms of PAM units, which represents the estimated number of mutations per 100 amino acids that have occurred during the evolutionary history of the two sequences that are compared. So to start with the substitutions or mutations, we have synonymous and non-synonymous mutations, which are type of point mutations that occur within the coding region of a gene, specifically within DNA sequence that codes for a protein, and these mutations can have different effects on resulting protein. So to start with the synonymous mutations, which typically involve substitutions of one nucleotide for another in the codon, but still the new codon codes for the same amino acid as the original codon. This is due to the redundancy of the genetic code, where multiple codons can code for the same amino acid. Synonymous mutations are often considered silent mutation because they do not result in any change to the protein primary structure or function. However, synonymous mutations can still have effects on gene expression, mRNA stability, and protein folding kinetics, as they influence the factors such as codon usage bias and mRNA secondary structure. Then we have non-synonymous mutations, which involve substitution of one nucleotide for another in a codon, leading to the translation of a different amino acid than what is originally specified by the wild type sequence. So these non-synonymous mutations can have various effects on the resulting protein depending on the specific amino acid change. These mutations can alter the protein structure, function, stability, activity, and interaction with the other molecules. Then we have the subpart of non-synonymous mutation is a nonsense mutation, which is again a type of point mutation that occurs in a coding region of DNA and results in the premature termination of a protein. This happens when a mutation creates a premature stop codon. That is, either the mutation makes the last codon has TAG, TGA, or TAA within the open reading frame of the gene. As a result of nonsense mutation, the translation of the mRNA transcript is prematurely terminated, leading to the production of truncated and usually non-function protein. Then we have missense mutations. Under missense mutation we have conservative and non-conservative mutation. So missense mutation, a conservative mutation in which amino acid substituted has similar properties to the original amino acid. So the similar chemical properties in the sense, for example, size, charge, or hydrophobicity. So conservative mutations often occur in regions of protein where maintaining a specific structural or functional characteristic is important. Despite the amino acid change, the overall structure function of the protein may remain relatively unchanged. Examples of conservative substitutions include replacing one amino acid one hydrophobic amino acid with the another, or replacing one polar amino acid with the another polar amino acid. Then we have non-conservative mutation, in which the substituted amino acid has different chemical properties from the original amino acid. So these non-conservative mutations can significantly alter protein structure, function, and stability, as amino acid change may disrupt important structural motif or functional domains. So these non-conservative mutations are more likely to be deleterious than conservative mutations, as they are more likely to disrupt protein function or stability. So to come back to our original topic, that is PAM matrix. PAM matrix is typically represented by 20 cross 20 matrix, where each cell represents the score assigned to the substitution one amino acid for another. The off-diagonal elements represents the scores for substitutions between different amino acids. So in these matrices, diagonal elements typically represents the log odd ratio of probability of a residue remaining. So the diagonal elements represents the log odd ratio of probability of a residue remaining unchanged over a specific evolutionary distance compared to the probability of substitution occurring. These values are derived from evolutionary considerations and statistical analysis. And in another way, the diagonal elements in PAM represents the score assigned to the matches between same amino acid in the alignment. It is important to note that the values may be vary depending upon the PAM model being used, which is PAM 1, PAM 100 and PAM 200, 250, as each model is constructed based on different evolutionary assumptions and data sets. The process of constructing a PAM matrix involves first step is the first step in constructing a PAM matrix is to collect a set of sequences that are closely related to each other. This is typically done by identifying a set of homologous from different species or strains. The next step is to align the sequences to identify the positions where residue substitutions have occurred. There are many algorithms and software tools that are available for sequence alignment, and the choice of method will depend upon the specific application. Once the sequences have been aligned, next step is to count the frequency of residue substitutions at each position in the alignment. This involves comparing the residues at each position in the alignment and keeping track of how one residue is substituted for another. Once the frequency of residue substitutions has been determined, the next step is to estimate the probability of each substitution occurring. This is typically done by dividing the number of times a particular substitution occurs by the total number of times the residue being replaced occur in the alignment. The final step in constructing a PAM matrix is to convert the probabilities of residue substitutions into a scoring matrix. This is done by assigning a score to each substitution based on the probability of the substitution occurring, and then normalizing the score so that the average score is 1 PAM unit in PAM 1. It is also worth noting that the construction of PAM matrix requires a large amount of data and careful statistical analysis. However, pre-computed PAM matrices are widely available and can be easily incorporated into the existing bioinformatics tools and pipelines. However, these PAM matrices are largely superseded by more accurate substitutions matrices such as BLOSSOM, which stands for block substitution matrix theories. Still, the PAM matrix remains a useful tool in some application and continues to be used by someone in research. So here, let's say we are studying about a protein X. So we collect the protein sequences of protein X from various species, human, mice and chicken. Then we align the sequences, and next step is to count the frequency of amino acid substitutions and then estimate the probability of each substitution and calculate the PAM score. So here I have given a PAM matrix, that is a pre-computed PAM matrix and alignment. So using this PAM matrix, which is a scoring matrix like PAM 50, can you try to score the alignment that is observed at the bottom of the screen? Score an alignment that is observed at the bottom of the screen using the PAM matrix provided. I got some answer. So this is the alignment given. So first we need to find out what is the score of T and Y in the matrix plus score of A and S in the matrix followed by score of H and D plus score of GG followed by score of KD. So here the score of T and phi in the matrix is minus 3 plus A and S is 1 followed by H and D is also again 1, GG is 5 and K and D is 0. So the overall score of the alignment is 4. So this is only based on the PAM matrix given. This is how alignment score is calculated using a PAM matrix. Higher the score.
Excuse me, sir.
Yes.
Excuse me, sir. Sir, can you please explain T and Y means exactly which two attributes? A N S H N D G G K D.
Yeah, just match with the T here and yes here. Sorry, T and
T and Y.
yeah. So T here and sorry Y here and T here.
Y on the Y axis.
Yeah, actually I have given a symmetric matrix kind of thing. Even if you build it the other way, this matrix look same.
Okay, sir.
So Y and T is minus 3 where my cursor is exactly.
Okay, sir.
Then A is here and S is in the second column, so it's 1.
Yes, sir. Got it, sir. Thank you.
Excuse me, I joined late. In the previous slide, can you please tell me what are the green highlighted ones and the ones which are not highlighted? What is the difference? What does this signify?
Yeah, so mostly the positive scores are highlighted. So here positive scores means it's identical substitution happening, and the red color digits which are highlighted in green indicates it's a score for similar substitution. That is, this is C score for C substituted over C, and all other positive scores are highlighted because these define identical nature.
So means positive versus positive or hydrophobic versus hydrophobic like that.
Yeah, so we can tell like positive scores in these alignments define identical residues in aligned positions. When you align in most of all these homologous or evolutionary distance, these are identical or conserved throughout. Negative scores are for mismatches mostly, which are not conserved. You see a lot of changes happening along the other family members. So where the scores vary.
Okay, thank you.
Next we have blossom. Similar to PAM, it is also used to quantify the similarity between the given sequences. So this blossom is based on the observation that some blocks of conserved residue substitutions occur more frequently than, than expected by chance in relative protein sequences. So previously it considered position based conservation or observed changes. Here some blocks, some window size of residues of conserved residue substitutions occur more frequently than expected by chance in related protein families. So the construction of blossom matrix starts by selecting a subset of sequences that are related to each other, and these can be obtained from a database of aligned proteins such as PDB or UniProt. Then we try to identify the blocks of conserved amino acid substitutions in the alignment. So these blocks are the regions of the alignment where there is high frequency of substitutions that are in the regions that are highly conserved across the protein families. Then we calculate the frequency of each amino acid substitution in each block. This gives a matrix of observed frequencies of amino acid substitutions. Then, like we did in PAM, we normalize the frequencies by the overall frequency of each amino acid in the protein family. So this gives a matrix of expected frequencies of amino acid substitutions. Finally, we then calculate the log-out ratio of each observed frequency to its corresponding expected frequency. This gives a matrix of substitution scores. So these are the basic scoring matrices that are behind dynamic programming concept. Then to start with the dynamic programming concept, so this dynamic programming concept is the extension to our sequence alignment topic that we have discussed in the last class. So in the last class we have discussed about the sequence alignment technique, that is dot matrix, where each the given two sequences are placed along the axis of a matrix and a dot is placed in a position of a matrix if both corresponding sequences have similar residues. And also this the dot matrix helps to identify the regions of similarity, the regions of repeats, the regions of inversion along with the palindromic sequences in the given two pairwise given two sorry in the alignment of the given two sequences. Followed by that, we also discussed about the limitations of dot matrices, as they are mostly sensitive towards the noise in sequences, followed by their limitations in handling the length of the proteins. So dynamic programming concept overcomes some of those limitations. So this dynamic programming is for solving optimization problems, particularly in sequence alignment and structural prediction. It is widely employed in applications such as sequence alignment, RNA folding, protein structure prediction, and hidden macro models. So here we have two main types of sequence alignment. One is global sequence alignment and local sequence alignment. In global sequence alignment, the goal is to align the entire length of two sequences. So we have algorithms such as Needleman-Wunsch that are used for global sequence alignment. It calculates an optimal alignment by considering all possible alignments and assigning scores based on match, mismatch, and gap penalties. The algorithm constructs a matrix where each cell represents the optimal score of aligning based on the prefix or score of the previous residue or previous alignment in that position. In this way, the matrix is filled iteratively considering all possible alignment chances. Then we have local alignment, where the goal is to identify most similar subsequences within the two sequences. We have algorithms such as Smith-Waterman that are used for local alignment. So to start with the steps in Needleman-Wunsch algorithm, so we have basic steps such as initiation, matrix filling, traceback. So initialization is creating a matrix with dimensions of n plus 1 cross m plus 1. So where n and m are the sequence length or sequence sizes or number of residues in the given two aligned sequences. So in this thing, first step is to initialize first row and columns of the matrix with the gap penalties. So this sets up the base cases for alignment. Then we have matrix filling. So for matrix filling, iterate to the matrix row by row from left to the right and top to the bottom, and at each cell in the matrix calculate a score of aligning. So to calculate the score, we consider three possibilities. The score of aligning characters at position I or j based on match or mismatch. Next, the score of introducing gap in the first sequence, aligning the second sequence character in the with a gap. Then we introduce a gap in the second sequence, aligning the character in the first sequence. Once we have three scores that are derived from the previous three cells of a matrix, we calculate the maximum of these three values as the current value score. Once matrix is done, it trace back from bottom right cell to the top left cell to reconstruct the optimal alignment. At each step, we make a trace back to determine whether the current cell value is obtained from match or mismatch, or a gap in the first sequence or a gap in the second sequence. Based on this determination, move to the neighboring cells accordingly and add the alignment characters or gaps to the signal. So now here I have started with the initialization step. So here I have used a gap score of -2. So initial we have aligned the sequences with the gap. So first step of initialization is assigning the first cell to 0 0, then aligning with the gap, aligning with the gap in the both directions. So for aligning with the gap, we just subtract with the gap score given. So 0 minus 2 is -2, -2 minus 2 is -4, -4 minus 2 is -6. So similar way, this has been initialization has been done, then followed by matrix filling. So the topmost image shows how a matrix is constructed and traceback arrows has been given. So these arrow marks indicate that the current score in the cell value is obtained from the previous cell values. So the current score cell value can be obtained from three ways. For example, the one value is maximum of three values that are observed by inserting the gap in sequence S or a gap in sequence D or a match or mismatch condition. So this arrow mark indicates that this one value is obtained from this 0. So it's a match or mismatch score here. So it's a match score here. So we will try this example to find the optimal alignment. So first step is I want to initialize the matrix. So I have initialized with 0 0, then with the gap. So the gap score is -3 here. So then we start with the matrix filling. So here I have constructed some part of the matrix, but the cell S4,2 is still empty. So I request all you to solve this cell S4,2 and obtain its value. So the value can be obtained, the formula is given here. Yeah, yes, please. So the given cell values are minus 11, 0, 14. So across the diagonal, try to use the match or mismatch condition, that is across the diagonal. So in vertical and horizontal lines, try to use the gap condition. Then take the maximum of 4 values. So the match score is given on top. So the match score is 8. Mismatch score is minus 5, that is considered as absolute value 5. Then the gap score is minus 3. Okay, first mention what is the condition here. If I consider diagonal, whether this cell has mismatch or a match condition.
Mismatch, sir.
Yeah, yes. So accordingly we take mismatch score. So minus 11, minus 5, 1 value. So minus 14, minus 3 is other value. 0, minus 3 is other value. So maximum of these 3 is the value for this cell. So this is how. So this s of x comma y depends upon match or mismatch. So since we all know one value for this is minus 11, minus 5, other value is minus 14, minus 3, other value is 0, minus 3. So maximum of these three values, which is minus 3. This is how a scoring cell has been filled. I have one more example as well. Yeah, now that we have discussed how to calculate position, yes. So why did I point an arrow mark towards 0 to 3, or in other ways people can point it from 3 to 2. So what does this signify? So this signifies this value is obtained from which of these three previous cells. So since minus 3 is obtained from 0th cell, that is insertion of a gap in the top sequence. So now that calculate the score for cell S5 comma 5. First make a note of its surrounding values. So again here the condition is mismatch. So this is the alignment. So we have values 19 minus 5 14, followed by 13 and 13. So the maximum value of these three is 14. So in that corresponding this cell, the value is 14.
Sir, for 19, the value 19, why it is minus 5, sir? It is matching, so shouldn't it be plus 8?
It is not matching. So here the corresponding cell value is T and it is C. It is a condition of mismatch. T here and C here.
Okay, sir. Got it. Thank you, sir.
So this way complete matrix filling is completed, and in the rightmost bottom cell it has been mentioned as a optimal score. So this is the final alignment score in terms of global alignment value. So if you're given this matrix, how a traceback and alignment has been constructed. So here I am highlighting the cells based on the color, how the current value has been derived given the based on the previous three values. So based on this I start from the bottommost right and the bottommost cell. So here AC and the previous value has been derived from diagonally. So either we consider it as a match or a mismatch situation. Then similarly it is also match or mismatch. So we align them similarly. Here we see the alignment started moving towards the sequence CA, CA, TT, GA in 16 to 19 sub. So we have inserted a gap here. So in between two T's we have inserted a gap. So if the diagonal line moves towards up, we'll try to insert a gap here. So this is how a optimal global alignment has been constructed. Now given this alignment, can you guys calculate what is the score of this alignment given the mismatch, a match and a gap score? Given the alignment, how what is the score of this alignment? Okay, if you have your constructed matrix here, as we discussed previously, this is the optimal score of alignment score. So the score of alignment of this, so the score for this alignment is 27. Let's see whether it's 27 or not, given the values. So we know the mismatch score is -5, match is 8, 8, 8, gap is -3, again 8, 8, and -5. So here totally we have 40 minus 13. So this is the optimal alignment score for this given two sequences.
Excuse me, what does this optimal score signify? Why do we need this optimal score? And just to know ki, which alignment is better, where the gap should come or something like that?
Yeah, so this is the maximum alignment score. Like this is the score between these two sequences. When you are comparing with other sequences, so higher the score, more similar are the two sequences. Or this is the best alignment that is possible between with a score of 27. So it's just like comparing. Higher the score, better is the more similar are the two sequences.
Okay, so does that mean when we align two sequences, so obviously there will be some mismatch or gaps. So the alignment which is giving the maximum score, we'll consider that.
Yeah, mostly if you are taking a pairwise sequence alignment, only one alignment is possible. What if I give you a third sequence and give now you need to make three pairs like sequence 1, sequence 2, sequence 2, sequence 3, and sequence 1 and sequence 3. Now if I tell which is the most closest two sequences, so whichever the two pairs is having higher alignment score are the more closest sequences. So that is multiple
Okay,
sequence alignment concept.
okay, okay. Thank you.
So still these calculations, this matrix filling, back tracing, everything is a complex job. So we have some softwares, that is emboss needle. So here using their this is the page for emboss needle. As we discussed, this algorithm is based on Needleman Wunsch method that we discussed just. So this method can be used to either align protein or DNA. So I have selected their example sequence. So we have used example option here. So the output format I want in pair format. Then I'm just giving trial 1. So just a couple of seconds the results will be displayed. So here the details has been given. So the program is needle. So here we have different types of penalties, that is gap open penalty, gap extend, gap end extend. So when we align two sequences, that is here I have aligned HPA human and HPA mouse sequences. The scoring matrices that I have used here is Blossom 62 with a gap penalty of 10 and extend penalty of 0.5. The length of the alignment is 142. So in this alignment of 142, we have 122 places where the sequences are identical and 131 places where the sequences are similar. So in remaining places there is a mismatch happening, and this alignment program has introduced zero gaps because of high gap penalty. So this is how the sequence alignment has been constructed using Needleman Wunsch algorithm. So this Needleman algorithm Wunsch is called global alignment because it aligns from residue 1 to the last residue of the sequence. So why it is called as a global alignment? It considers whole protein in the alignment process. So instead of handling all this matrix, so this algorithm provides the direct alignment as a output. Further, if we want to try our hands on, so this is the code. I'll share the link. So I have shared the link. So one can copy the link, copy link address, then go to your new tab, any of your browser, internet browser, paste it, enter. Then open with Google Collaborator. So using what all the steps we have discussed, I have made a function. So this
Excuse me, please share the link again. I just there was a connection issue, so I missed that.
okay, okay, I'll share it again. So given any function, we need to specify what input the function needs to handle. So here I have specified two sequences. Since this is a pairwise method, I have given two sequence 1 and sequence 2 as output, followed by match score, mismatch score, and a gap penalty. So using these three types of scores and two sequences, this function will calculate the global alignment. So first I initialize the matrix with the 0 in the leftmost and the left and the topmost cell, followed by I have made a matrix for trace back. This is for the last step. Then here I have initialized the first row and column of the scoring matrix. So this first row and the column of the scoring matrix is initialized by aligning it with the gap. So here we don't consider any match or mismatch, directly align them with the gap penalty. Similarly, first column I have aligned with the gap penalty. Then I started with the filling the scoring matrices. So this filling of the scoring matrices is based on the maximum value obtained from match or mismatch, or obtained from gap penalty from vertical or horizontal cells. So finally when I get three scores, that is a score from match or mismatch, or a gap penalty score from horizontal cell and a gap penalty from vertical cell, the maximum of three values is considered as the final score. And then finally I am saving the final score in the matrix. So finally I have generated a trace back matrix here. Here I am giving the similar example that we have solved. So as I mentioned in previous class as well, there is this play button. So if you click on this, the cell will get run. when you run the code, outputs the scoring matrix. So this scoring matrix is similar to the scoring matrix what you have seen in the slide, with an optimal score of 27, and this is the alignment with a gap introduced in the sequence 2, that is the sequence starting with CAA, and the alignment score is 27. So further, users can change the sequence here and the alignment will be changed, and based on the requirement the match score and mismatch score and gap penalty can be reduced. If you don't want gaps in your sequence, keep a high penalty for the gaps so that the program itself try to avoid adding gaps into the final alignment. So this is how a global alignment is done. Then I have local alignment, which is mostly the similar concept. So in this Smith-Waterman algorithm, which performs local sequence alignment, this is used for determining similar regions between two strings of nucleic acid sequences or protein. So this local alignment, instead of looking the entire sequence, it try to find the subsequences that match with each other. Previously in global alignment we have maximum of 3 conditions. Here we have extra conditions, that is maximum of match or mismatch score, maximum of gap penalty from vertical and horizontal cells, followed by maximum of 0. So in this scenario, since we are considering 0, maximum of 0 is included, the minimum value that we can expect in these is 0. So no negative values are observed in Smith-Waterman algorithm. So here similarly the same alignment, I try to generate a local alignment. So in the first step is to initialize with 0 and 0. Then all this has been aligned with a gap. So since the gap penalty is -2, we have a condition maximum of 0 comma -3. So the first two cells are filled with 0. No negative values are observed in local alignment. So in this way I have filled a matrix. Now try to find what is this value significant. What is this value? So in the
With 7?
yeah, yes please, yeah. So with the exam point of view, you can expect the questions in exam, uh, similar types of questions, like given a half filled matrix, we expect you to find the value of a particular cell. So similarly we need to write what are the previous cell values, 5, 3, 10. So what is the condition here? The condition is match or mismatch.
Mismatch.
Yeah, so we need to subtract with -5. So 5 minus 5 we have 0. Then 3 minus 3 0. 10 minus 3 7. And here the fourth condition is 0. We should include 0. So maximum of these three. So the answer should be 7. So first we need to trace back with the arrows. Then the value is 7. So first I have made the alignment like this. So in global alignment, we have only three conditions. If we consider global is 0, 0 and 7. If we take local alignment, this is how the construction happens. We have fourth condition, that is include 0, maximum of match or mismatch score, gap penalty from vertical cell, gap penalty from horizontal cell, followed by maximum of 0. So this is the small change that both algorithms have. I hope everybody got this score.
Is there any online software for this matrix also, like you told for the previous one, emboss needle?
Yeah, we have emboss water. I'll just show you in a couple of minutes. I have a question here. How many of you have used Blast? Any Blast? Blast P, Blast N? Yeah, so this is the background. Like we can tell, it's a raw background behind the working of Blast. So Blast search is basic local alignment search tool. So why when we observe a Blast result, we see coverage is also important. If coverage is less, that means Blast is searching for subsequences as well. Uh, Here I have a question. So coverage is in what percentage of the given input sequence 1 is matching with the sequence 2, is aligned with the sequence 2. In local alignment, what can we sorry, in global alignment, what could be the coverage, sequence coverage? So sequence coverage is given an alignment, number of residues aligned from sequence 1 divided by number of residues aligned by sequence 2 into 100. So what could be the coverage score we can expect in global alignment? So when we are aligning from one corner of the sequence to the other corner of the sequence, covering whole the sequence, so the coverage will be 100 in global alignment, and we consider blast, the coverage will be less than 100. So similarly, we have a traceback concept, but the traceback concept doesn't start from the right and bottommost cell. It will start with the cell that has the maximum value. So here 37 is the cell that has maximum value and ends with 0. So this is the alignment matrix that you have seen. So 37 is the maximum, and here in 37 we are moving diagonally, so it's a match or mismatch condition. So GG has been aligned, then again we move diagonally, so TT has been aligned, the corresponding cell values has been aligned, then we try to move towards the gap penalty, so a gap has been introduced here, so the C value here has been introduced with the gap, then again we move diagonally, so T and T has been matched, A and A diagonal movement, A and A is a diagonal movement. So this is finding the similar subsequences that are available in the given input sequence. Just
So in this, the last residue will not be shown.
uh, last residue in the sense, sorry, I couldn't get. Yeah, you mean the 0 0?
No, no, the last one, like you ended till 37.
So after 37 means this G is a perfect match. After that there is a C and A, matlab C in one sequence and A in another sequence. So that is not to be shown as a mismatch or a gap.
Yeah, so it won't show unless the score has a positive value. If the score is 0, the local alignment stops there.
I'm talking about the last one. Yeah, okay, maybe
Yeah, so this is not considered because the highest score starts from highest score. Local alignment starts with, irrespective of high or low, irrespective of high or low values. This starts with the from one corner of the matrix to the other corner. This is global alignment. Here we have 32, but in global alignment we won't consider 32. Whatever the value we see in the last cell we consider, but in local alignment we consider cell having the maximum value and trace back towards the up, towards this 0. We trace back towards this 0. So this is the
okay.
difference between both the alignment techniques. So here
So
37 is
both
the maximum
uh,
value. Sorry.
both the alignments will trace back to 0, but one's will start from the last value and the other one, the local one, will start from the maximum value.
Maximum value. Yeah. So similar to embossed needle, we have embossed water. Okay, I'll just paste the link in the chat box as well. So both the interfaces look the same because these both are handled by EBI website, European Bioinformatics Institute website. Then similarly, I want to use the example. Title is trial 2 and submit the job. Yeah, the details of the algorithm has been given before the alignment of the sequence, followed by the alignment of the sequence. Yeah, so there is a question that has been asked about the query coverage. Okay, I'll take these two examples. Just give me a minute. So here on the screen, whatever you see is, let's consider CAA has our input sequence which starts with CAA T underscore TGA has our query sequence. So when we aligned with the next sequence, that is GAA TCT, so let's see in final alignment how many number of residues has been aligned. So this is our query sequence. So the length of the sequence in this alignment is 8 divided by what is the other length of the sequence that has been aligned. So here also we have 8 residues that have aligned. 8 into 100. So length of the aligned query sequence followed by length of the aligned subject sequence into 100 gives the query coverage. So this is 100%.
Sir, one gap is there, so
This is considered as the final alignment. So in global, gap is also scored, so it is also counted.
okay, sir. Thank you, sir.
So when you go for a local alignment here, the number of sequences residues that are aligned from query sequence divide by total length of the query sequence. That is, here the query sequence length is this 8. Still the query sequence length is not changing here, but sorry, the subject length is not changing, but the query aligned length is changing to 6. So 6 divided by 8 into 100. So this is the alignment score that is given for local alignments.
Sir,
Yes.
for local alignment, is it like 6 by 8 into 100, and for global it is 8 by 8 into 100? It's like that.
Yeah, for local it will vary. In global alignments, the coverage is 100%. It's not like values, because we try to align whole length of the sequence, whole length of the sequence we try to align. So the coverage will be 100%. Like
Okay.
100% of the query is covered, not with respect to the subject. It's like again.
Okay. Thank you, sir.
But whatever the scores we see in BLAST are based on local alignment search. So I'll start with the BLAST and FASTA, which are widely
Gap
available.
is, sir, so gap is considered only in local alignment.
In whole alignment concept only, the gap is considered to calculate the coverage.
Okay.
Only in terms of similarity identical scores, the gaps are not considered.
Okay, sir. Thank you, sir.
Yeah, yes. By default in global alignment, the coverage is 100% if we are considering gaps as alignments. And this the align if you don't consider the gaps as alignment, the alignment score will change. I'm answering to a question in the chat box. So now we have BLAST. So this BLAST stands for Basic Local Alignment Search Tool. So it is widely used for comparing biological sequences and used to search similarities between query sequence and sequences in the database. So the key points about BLAST is it is an heuristic word based method where the BLAST uses heuristic word method for fast pairwise sequence alignment. This method involves the breaking sequences into smaller fragments and comparing them to quickly identify potential regions of similarity. And we have different versions of the BLAST, so which are developed for specific types of sequences and databases. Firstly, we have BLAST 10, which is used for nucleotide sequence search against the nucleotide sequence database. Then we have BLAST P or protein BLAST, which is used for protein sequence search against the protein sequence databases. Then we have BLAST X, which is used for translating nucleotide sequences in all 6 reading frames and searching them against the protein sequence database. Then we have tBLAST N, used for translating protein sequences in all reading frames and searching against a nucleotide sequence database. And finally, we have tBLAST X, which is used for translating both query and database sequences into 6 reading frames and performing a comparison. So this BLAST uses a heuristic word search method which rapidly tries to identify local alignments between the sequences. These strategies involve efficiently searching the databases for high scoring segment space, that is HSPs, based on sequence similarities. So the other one is FASTA, which is this FASTA is also different from FASTA format, which is another popular tool for sequence alignment and used to search similarities between biological sequences. So FASTA is primarily used for uh, sequence alignment, comparing a query sequence to a sequences in databases to identify similarities. Similar to BLAST, FASTA also employs heuristic word method for fast sequence alignment. So FASTA is mainly used for tasks such as sequence homology searching, sequence database searching, or motif discovery. And finally, the extension version of this pairwise sequence alignment is multiple sequence alignment, so which is a method for aligning 3 or more biological sequences to identify conserved regions or evolutionary relationships. So MSA is a weak 4 concept Now I would like to move on to the quiz. So the first question is, the gaps are added to the alignment because they increase matching of identical amino acids at subsequent portions in the alignment, or increases the matching of dissimilar amino acids at subsequent portions in the alignment, reduces the overall score, or enhances the area of the sequences. So why a gaps has been in other terms, the gaps may also be termed as insertions or deletions or in a given sequence. So the gaps has been introduced so that we have high matching of identical amino acids or high matching of dissimilar amino acids, or to reduce the overall score or to enhance the area of the sequence. Yeah, so the gaps has been added so that they will increase the matching of identical amino acids at subsequent portions in the given alignment. So these are some of the reasons. So gaps allow for better alignment accuracy and also reflect evolutionary relationships and facilitate functional prediction and guide in experimental design. So dynamic programming involves processes. So this is like words filling. So these are the steps in dynamic programming. So what is option 1? So it's like form a proper word, second one, third and fourth. Okay, I have an answer for the first one. So the first step is initialization. So where we give 0 to the left and topmost cell, followed by initializing with the gap. Then what is the second one? What is the second step we do? So we initialize the matrix, then we fill the matrix.
Find the match and mismatch.
Yeah, it's like the step is mentioned as matrix filling. Yeah, thank you for the answer. Okay, once we fill the matrix, so what is the option 3? What we do once the matrix is completed?
Maximum score is found
Yes,
out
then
of the three, from the three scores.
yeah, then after that, once whole matrix filling is done, so let's go back and see the steps again. So we have initialization step, sorry, initialization step followed by matrix filling. Once whole matrix filling is done, we do trace back. Once trace back is done, then we have alignment. So the option 4 is final one, that is aligning. So we initialize the matrix, then we fill the matrix, we do trace back, and finally obtaining the alignment or aligning the sequences. So these are the 4 steps that are involved in a dynamic programming process, mainly in Needleman-Wunsch algorithm and Smith-Waterman algorithm. So given in what alignment two sequences are to be aligned are assumed to be generally similar over entire length, and in what alignment on other hand does not assume that two sequences in question have similarity over the entire length. So in what alignment we consider whole similarity over the whole length of the sequence, and in what alignment we does not assume that the sequences are similar identical over the whole length. So here this question asks for two answers. I'll wait for some other answers. So the alignment in which two sequences are aligned are assumed to be generally similar over the entire length is global. So it's end-to-end alignment, and the alignment which does not assume similarity over the entire length is global alignment. So given we have discussed about the local alignment step, which of the following does not describe the local alignment algorithm? Which does not describe the local alignment algorithm? So local algorithm can have scores be negative, or negative score is set to 0. First row, first column are set to 0 in the initialization step. In the traceback step, it begins with the highest score and ends when 0 is encountered. Which does not describe. So in local alignment we don't have negative scores because we consider a fourth condition, that is maximum of mismatch or match score, maximum of gap penalty from horizontal cell, or score of gap penalty from vertical cell, and 0. So maximum of three values. So the minimum value that one can expect in a local alignment algorithm or local alignment matrix is 0. Minimum value that one can observe. So scores can never be negative. So the quality and time for database searching depends on size of the query sequence, number of sequences in the database, or both A and B, or none of the above. Yeah. So when we consider the given example of local alignment only, pairwise alignment, the matrix size is directly proportional to the query size. So if the query size is big, the matrix is big. So the time taken to construct the matrix is big, and also given in a database, all possible combination of pairs with a given query sequence will be generated, and the alignment has to be constructed for each pair. So the quality and time for searching a database depends upon size of the query, that is query's input sequence and number of sequences in the database. If in if input query is large, time taken is more and quality reduces, and number of sequences in the database is many, irrespective of query size, the time taken is high because it has to generate pairwise alignment with it's a query, and each sequence pairwise alignment will take time. Then, yeah, which of the five basic BLAST programs you should use if you have a protein sequence and wish to know what other proteins look like it? So for reference, I give you a table initially. So if you have a protein sequence and you wish to know what other proteins look like it, so what BLAST program one should use? Yeah, it is BLAST P, protein BLAST, where if we have a protein sequence and we wish to know what other proteins look like it, so it is protein BLAST. Then if you have a DNA sequence and you wish to search for other DNA sequence to find that encodes the similar protein is, so I give you a table. So you have DNA sequence and you want to search for a DNA sequence. So it is DNA to DNA. So it's just sorry, I think I missed up with some answers. Yeah, so since we have that encodes the same or similar protein, so you need to translate the given DNA sequence and then translate it back to the DNA. So it is tBLASTX, because there is one extension that find one that encodes the same or similar protein. So this is more simplified version of the previous question. So this is nucleotide BLAST 10. So we have a version of BLAST where the given DNA is translated into 6 frames and the DNA sequences in the database were also translated to 6 frames, that is 6 protein reading frames, and then try to find the same or similar proteins, that is T blast X. And we have other DNA sequence, other blast version, which is used for only directly mapping DNA with DNA sequences in the database. Then ninth one. So we have protein sequence and we wanted to search for DNA databases to find genes. So this is tblastn, where the query sequence is protein and database to search against is DNA, which is translated into 6 reading frames. Then this is the last question for the day. So BLAST uses what to find matching words, and whereas FASTA identifies identical matching words using so BLAST uses substitution matrix, whereas FASTA uses hashing position. I have one more. So given two types of sequence alignment, so what does the first one and the second one denote?
Sir, what is the correct answer of the previous question?
It was substitution matrix and
Okay,
hashing position.
thank
So BLAST is local alignment, so which is matrix based scoring. This is hashing position. It's like masking and finding out the words. So the first one is global alignment. The next one is the local alignment. Yeah, that's all from my side. Any questions? If no other questions, you are free to leave the class. See you all next week. Thank you for attending. If any questions, I'll be happy to take those questions.